Good day, everybody. In today's lecture, we'll first go over organic chemistry, and then we'll go ahead and explore the composition, structure, function of some large biomolecules, such as proteins, carbohydrates, lipids, and a nucleic acid. Before we go on, I would like to give you a disclaimer. Because of the vast amount of information we have to cover for this chapter, we are going to only scratch the surface of this topic. First, we'll talk about some molecular structure and the functional groups of organic molecules. And then we'll go ahead and talk about the relationships of different organic molecules. In that section, we'll go over the concept of different isomers. For example, constitutional isomers versus stereoisomers, etc. Then next, we'll talk about some biomolecules. First, we'll talk about proteins, and we'll talk about the protein structure and function, and followed by carbohydrates, lipids, and nucleic acid. So let's talk about organic chemistry first. So what is organic chemistry? Organic chemistry is the chemistry that is based on carbon. So what about the biochemistry? Biochemistry is the study of a chemical process taking place in the living organisms. Then biomolecules, they are usually also organic molecules, but those molecules are produced by a living organism. So what is special about carbon? Why is carbon the selected element for the foundation of organic chemistry? The reason is because carbon is very versatile. First, carbon is tetravalent, which means carbon can form 4 single bonds surrounding itself, or equivalent of that. For example, in the first example, you see 4 single bonds surrounding the carbon. The second one, you have a double, and you have 2 single. The third, you have a double-double combination, and the last one, single and triple combination. In this way, it creates very diverse geometries around the carbon center. For example, when you look at carbon number 1, 4 electron groups, 4 single bonds, 4 electron groups, with no lone pairs around the carbon. This gives a tetrahedral geometry, and the hybridization type is going to be 4 electron groups, so it's sp3 hybridization. For the bond angle, you look at the bond angle, notice that is not 90 degrees because this is in a tetrahedral shape rather than a square planar shape. So this is actually 109.5 degrees C. For the second one, similarly, the carbon has 1 double bond, 2 single bonds with no lone pairs surrounding that carbon. That means it's 3 electron groups. The geometry for the carbon is trigonoplanar. The hybridization would be sp2. Bond angle would be close to 120. It should be slightly smaller than 120 because the double single bond repulsion should be slightly bigger than that between the 2 single bonds. For the third one, the carbon has 2 electron groups, one on the left, one on the right, for both the third and the last one. So carbon would have a linear shape. The same for the last one. It's also linear. Okay? All right. 2 electron groups surrounding that carbon. So it's sp hybridization. Bond angle, it will be 180 degree for both of them. Another source of the diversity or versatility of carbon comes from its ability to catenate. So what is catenate? Catenate means the ability for the element to bond to the same element. So for carbon, it can bond to other carbons and form different shapes, form chains, branch chains, or rings. As you can see, that's a chain, branch chain, and the rings over here. Third reason, carbon can actually form very stable bonds with many other elements. For example, hydrogen, oxygen, nitrogen, halogen, and some other elements. To be clear, for later discussion, heteroatoms are defined as those atoms that are not carbon or hydrogens. Let's give you some examples about how organic molecules are represented. We have different ways to represent it. For example, for this compound, carbon, 4 carbons are chained together. All right. Then you might be wondering, what else besides this carbon? For organic chemistry, we do notice one thing that is very important. We said carbon is tetravalent. So for the first carbon, you only get 1 single bond surrounding it. So you have 3 spaces to fill. You use hydrogens to do that. For the second carbon, you have 2 single bonds, one on the left, one on the right. So you have 4 total, so you need to put in 2 hydrogens. The third carbon is the same way. You need 2 more hydrogens to satisfy the tetravalency for carbon. The terminal carbon on the right side, you need 3 more hydrogens. So this is one way to represent the structure. And for this structure, it's very straightforward with all connectivity clearly indicated. And we call this type of structure structural formula, because it does clearly indicate all the details about the structure. But this does take a long time to write it. So another way to write it is to group all the hydrogens on each carbon. For example, for the first carbon, you have 3 hydrogens on it. The second carbon, you have 2 hydrogens on it. Then third one, look at, you have 1, 2. So if we group the hydrogens on each carbon, we can write it this way: CH3, CH2, CH2, and CH3. So if you group the hydrogens on each carbon, we call this condensed formula, or some textbook might call that a condensed structural formula. I'm going to just call this condensed formulas. Okay. Right. The third way to write it is to count all the elements of the same type. For example, in this compound, you have 1, 2, 3, 4. 4 different carbons. So with all 4 carbons together, if you count the hydrogens, you would come to 10 hydrogens. If you put this together, then this formula is so-called a chemical formula or molecular formula. Another way, or the last but not the least, we're going to introduce to you is so-called line structure. In this structure, we use zigzag lines to represent the backbone of the carbon. So at the terminal position, we have 1, look at this, so 1, 2, 3, 4. So you look in this line structure, or sometimes it's called a bond line structure or bond line structure. Okay, but I'm going to say this, called a line structure. In this line structure, first you notice the corner or the terminal position. Okay, they are indicating carbons with certain number of hydrogens. Okay, how many hydrogens? It depends on the tetravalency, right? So for example, when you look at the top one, the first one, that's a carbon. You look at this corner, that's a carbon, carbon, carbon. So you can actually go back to the original. For example, that's my carbon. At the terminal, no, that position is a carbon. That's another carbon. So this is carbon. Okay, carbons are not indicated. Hydrogens are not either. So you're going to put back the hydrogens. So this carbon needs 3 more hydrogen because it's only bonded to one single bond. For the second one, you're going to put 2 hydrogens to it. Similarly, for this carbon, you need 2 more. The last hydrogen, you would need 1, 2, and 3 hydrogens. Okay, so this is actually the structural formula we just rendered from the line structure. What is slightly different from the original one we have over the top is now, rather than doing the horizontal way, we have zigzagging way to do it. But it's also the complete picture of how the atoms are connected. So let's just to make it 100% clear, the carbons and the hydrogens. So in the line structure, the carbons and hydrogens, okay, they are not indicated. Okay, except when hydrogen is attached to heteroatoms. So hydrogens on heteroatoms, they are indicated. So we just mentioned the heteroatoms are the atoms that is neither a carbon nor a hydrogen. So they are indicated. Okay. Let's go over a few more examples for better understanding of these different representations. For example, look at this structure that is indicated over here. This structure is the line structure, as you can see. Can you write the other 3 different representations based on this structure? We can, because we know from the line structure, the corners or terminals, they are the carbons with certain number of hydrogens, but they are not indicated. The heteroatom, as you can see, the oxygen over here is indicated. If you have hydrogens on heteroatoms, they should be indicated as well. Right. So look at the structure. We can replace the original corner or terminal with carbons. So that's my carbon, and then the next one, that's carbon, 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 carbon. All right. Then it's connected to another carbon. We're going to write it almost exact the same way as the line structure, so you can see the correlation. Then we're going to fill in the number of hydrogens. We do know carbon is tetravalent, so we should make sure carbon has 4 single bonds or equivalent of that. So we do know we can fill in the correct number of hydrogens, as I am doing right now. For this hydrogen, for this carbon, it only has 1 more space for 1 hydrogen. Then when you look at the hydrogen that is connected to this oxygen, it has 4 bonds already, 1 double, equivalent of 4 single bonds already, 1 double bond and 2 single bonds over here. All right, then let's keep going. So here, this carbon needs 2 more here as well. Then this terminal one would need 3 more hydrogens. So as you can see, it takes up a lot more space and takes a lot more time to complete the structure. This one is called a structural formula. This gives you the complete picture about how all atoms are connected to each other. Right? If we try to condense the structure a little bit, which means you will write the hydrogens on each carbon together. So if you write it that way, it would be, so here would be CH2, now a CH2, and CH2, CH2, CH2. This is carbon then. Now, for the connection, this is actually not CH, this is CH, right? Okay, now CH2 for this one, then CH2, CH2, CH3. That's the condensed formula. It also takes quite a bit of time. Okay, if you write the whole formula, you can count the number of carbons and the hydrogens. You can also write the chemical formula. All right, we're gonna skip that step. Let's go over another example. For example, if we have a structure like this, can you write other representations as well? Okay. You look at any corner or terminal position, these are carbons. And this is also carbon. Right? And NH3, NH2, when you look at that, the hydrogens on nitrogen, which is a heteroatom, the hydrogens on nitrogen are indicated. You see that? You cannot just simply write an N because it is a heteroatom. It's required to write the hydrogens on heteroatoms. So let's convert this to corresponding representations. So you look at here, the terminal ones. So this one is not carbon because it is connected to NH2. You see that? So this one, rather than writing the carbon, you should write N with two hydrogens. So one maybe right on the top, one on the right side. Okay. The other hydrogens, carbons, we're going to write hydrogens surrounding that. All right. Hopefully this helps. If you want to write the condensed formula, you can write it as, let's write the center carbon first. Then the top you have a CH3. The left you have CH3. Notice the carbon is connected to the neighboring carbon, right? Not the hydrogen that is of one carbon is connected to the next carbon. No, okay. CH3. Then NH2. That's the condensed formula. Okay. You can also count all carbons, hydrogens, and nitrogen. Then you have chemical formula. Right. The chemical formula for this one would be 4 carbon, 11 hydrogens, and the 1 nitrogen. That's the chemical formula. What about the bond angle and hybridization? For example, when you look at this carbon, what is the hybridization? When you look at the hybridization for a large molecule, like you see over here, only look at that particular center. So for this carbon, you have one double bond, okay, one double bond, and 2 single bonds connect to it with no lone pairs over here. So you have 3 electron groups. The hybridization would be sp2. The bond angle would be 120 for ideal situation. So it's slightly smaller than 120 because you have a double bond. Double bond repulsion between double and single is slightly greater than that of single and single. So that's why it should be slightly smaller than 120. All right. Then you might be asking, what about this carbon? What's the hybridization? When you look at hybridization for that carbon, it has 4 single bonds and no lone pairs. In total, it has 4 electron groups surrounding that carbon. So it will be sp3. If you're not clear about this concept, please review the Lewis structure of General Chemistry 1. Okay, all right. So the bond angle, rather than 90 degrees, it will be 109.5 degree for tetrahedral shape. Okay, right. So far, we have spent a lot of time talking about the organic molecules per se. Now let's talk about how to classify different organic molecules. We classify organic molecules by looking at so-called functional groups, which tells you what type of chemical reactions they can undergo when you look at the corresponding portion of the substance. For alkane, they are all carbon-carbon single bonds or carbon-hydrogen single bonds. We usually say they don't have a functional group, but we write it over here so you can see. That's why you don't see a highlighted area. For alkene, which has ene ending, you will always see carbon-carbon or CC double bond. For alkyne, it means somewhere you will see CC triple bond. For so-called alcohol, for example, ethanol, right, or methanol, as you see over here, you always see a hydroxy group. What do you mean hydroxy? That means OH group. The difference between the hydroxy group and the hydroxide is hydroxy is actually neutral, OH. No charge on it. Hydroxide is OH minus. What is carboxylic acid? Carboxylic acid, it has both the CO double bond connected with OH. Okay, so it's not only it has a CO double bond, okay, it also has an OH. You cannot single this out, OH, and you call that alcohol. That's alcohol function group. That's wrong. If you have an OH group connected to CO double bond through that carbon, the whole thing is carboxylic acid functional group. You cannot separate it. Okay. All right. What about amine? Amine is the derivatives from NH3. Basically, for NH3, you have 3 hydrogens, but up to 3 of those hydrogens can be replaced with carbon chains, which is indicated as R over here. That means carbon with certain number of hydrogens on it. Okay. So for example, methylamine, the NH2 will be so-called a functional group for amine. Aldehyde. Aldehyde has a carbonyl group. Carbonyl means it's carbon connected to oxygen with a double bond. But it's not the only thing. It's also connected to hydrogen. Okay, so it's the carbonyl group connected to that hydrogen that makes up the aldehyde functional group. Let's look at next one, ketone. As you noticed, the CO double bond itself does not clarify which kind of a functional group this is. As we can see in previous examples, hey, look at carboxylic acid. This guy has a CO double bond, and aldehyde, you have a CO double bond. When you look at ketone, it also has a CO double bond or carbonyl group. Okay, so the carbonyl group by itself does not tell you specifically what functional group that is. It is combined with how these groups are connected with the surroundings. Then you know what kind of functional group this is. For example, for carboxylic acid, that carbonyl group, this carbonyl group, as you can see, is connected to OH on one end. Okay. Then that's carboxylic acid. For aldehyde, it's the carbonyl group connected to hydrogen. Then you call that an aldehyde. For ketone, it's the carbonyl group connected to two, you see the R or R prime. That means they are carbon chains. Carbon connected with certain number of hydrogens. They are so-called alkyl groups, by the way. All right. So the R or R prime, they are alkyl groups, which means they are carbons or carbon chains connected with certain number of hydrogens. So, for example, in acetone, you have a carbonyl group which are connected to carbons. You see that? They're connected to carbons. But how many hydrogens are connected to it? Since you only have one carbon, that's why you have 3 hydrogens. On the left side, it's also the same. Remember, if you replace CH3 with hydrogen, that would make this compound an aldehyde rather than ketone. Remember, for example, if I write something like this, that would be an aldehyde. Why? Because that's a carbonyl connected to hydrogen. So this whole thing, as you can see in this large circle, that's an aldehyde functional group. All right. For aromatic, you will have a ring which has alternating double bonds in it, as you can see over here. This is benzene. All right. What about ether? Ether contains oxygen, but as you can see, many compounds, organic compounds, have oxygen. Like you see in, hey, that's oxygen. Look at the previous. Oh, you see oxygen. You see oxygen, oxygen, oxygen. So what I'm emphasizing here is oxygen by itself does not make an ether out of it. It's the oxygen that is flanked by 2 R groups or 2 carbon chains, carbons with certain number of hydrogens. That makes oxygen an ether functional group. As you can see in dimethyl ether over here, you have this oxygen is flanked by 2 carbons which are connected with correct number of hydrogens. In this example, that's dimethyl ether. All right. S2 has a carbonyl group and is connected to 1 oxygen with a single bond, and the R prime is certain number of carbons, and this is the ester functional group, as you can see over here. This carbon, as indicated by this arrow, is quite important. If that CH3 is replaced, remember, if that CH3 group is replaced with hydrogen, that functional group will not be ester, rather it will be carboxylic acid. Okay, this is quite complicated, I know. Yeah, please spend some time and study on the difference for different functional groups. For amide, that's the CO double bond or carbonyl connected to NH2 or N connected with certain number of carbons. That will also work. So carbonyl connected to nitrogen, that's so-called amide. Some textbook call, some people pronounce this as amide. Usually I prefer amide. Let's summarize everything about functional groups. First, functional groups indicates the type of organic reaction this type of compounds can undergo or can usually undergo. For alkane, it does not have a functional group. Carbon are connected with a single bond. Carbon and hydrogen, a single bond as well. If you have CC double bond, that's alkene. CC triple bond, that's alkyne. And if you have an alcohol, that means you do have an OH group. But again, as I emphasized many times, OH group itself does not guarantee an alcohol. It's OH group that is connected to this R, which means a carbon chain, also called alkyl group. What do you mean alkyl? Alkyl group. Carbons with certain correct number of hydrogens. Okay. Do remember the surrounding of the functional group is important. So that's it about the functional group. Now let's continue and talk about another concept, isomers. So what is isomers? When you get isomers, isomers means, isomers means the compounds with the same formula. Okay, same formula, but different structure. Okay, right. And when you talk about isomers, we have different types of isomers. One is called a constitutional isomer. One is called a stereoisomer, which we'll go over later on. Constitutional isomers are the molecules that have the same molecule but have different connections between atoms. I'm going to give you one example. For example, you have this compound. That's butane. We went over this previously. So when you write the formulas for butane, this would give you C4H10. We have another compound that has 3 carbons in the main chain, and you move the next carbon like this. So when you write the hydrogens, okay, so if you write the hydrogens, and then you do the formula, you will notice that it has exactly the same formula, C4H10. But apparently, when you compare these two formulas, they have different connectivity. The carbons are connected in different ways, and some hydrogens are connected in different ways as well. So these two isomers are called constitutional isomers because the connectivity between atoms are different. How do we determine if one structure and another, they are isomers, or are they identical? So here's how to do it. For example, in structure 1 and 2, when you look at that, they might look like they are constitutional isomers, but they are not. Because when you rotate it, as you can see on the right side, rotate this thing 180 degrees, they become exactly the same. For simple cases, we can do rotation if they become the same, so we know they're identical rather than they are isomers or constitutional isomers. Okay. The second method is to number the carbons on the structure. For example, for a more complicated case, as you see in structure 3, we can number the carbons on the longest carbon chain. Okay, longest carbon chain as highlighted on the left side. Okay, so we number this as 1 because if you start 1 on the left side, the first branch, which is not part of the main chain, will not have the lowest possible number. So we're going to make sure the branches. What do you mean branches? Branches means if it's not a part of the main chain. All right, the main chain is continuous carbon chain. It cannot be, you cannot just jump around. It must be continuous. Okay, so the longest chain, you number carbon 1, 2, 3, 4, 5. Then this branch as I circle, and that branch, they will have the lowest possible position numbers. All right. So then you will look at the structure 4. Seemingly they are different, but if you number the main chain, you will notice, you see, on carbon 2, that's carbon 2, you have 1 branch, and on carbon 3, you have another branch, which happens to be exact the same as structure 3. So structures 3 and 4 are identical. They are not isomers. So far, we have learned what constitutional isomers are and how to differentiate identical structures from isomers, from constitutional isomers. Now we're going to go on and talk about the second type of isomers, which is called stereoisomers. Stereoisomers, of course, they're isomers, so they are also going to have the same formulas. But do remember, the connectivities are also the same. The difference for stereoisomers is the spatial arrangement of the atoms, or 3D arrangements of the atoms. For the stereoisomers, we have many different ways to further classify that. You might have so-called cis isomer or trans isomer, and enantiomers and the stereoisomers. Okay, we're going to go over this one by one. All right. So you look at examples over here between 1 and 2. Okay, between A and B actually. So when you look at A structure here, and then you look at on the right side. Okay, apparently you look at the carbons 1, 2, 3, 4, 5. Look at all the carbons 1, 2, 3, 4, 5. All carbons are connected exactly the same way, and the hydrogens they are grouped on the carbon. When you look at that, they are also connected in the same way. So in this one, as you can see, the connectivities of the atoms are also the same. So what's the difference? The difference is how they are arranged. You look at this double bond here, okay? When you look at that double bond, and you do know if you want to write the hydrogens here, that would be another hydrogen. On this side, that's also hydrogen. But then for the structure on the right side, that's where the hydrogens are placed. So on the left side, the two hydrogens on two carbons of the double bond that form the double bond, they are on the same side. If it's on the same side, okay, for the two hydrogens, it's actually two hydrogens which are the same. They are on the same side. Then we call this cis. That's the reason, because you have the same groups on two different carbons on the same side. Then we call this cis. If the same group on different carbons that form the double bond are on different side, we call that trans. So for trans, it's hydrogens. They are on opposite side. Okay. If you write the more detailed, the condensed formula, as you see at the bottom, as you can see more clearly the connectivity between the carbons and the hydrogens are the same, or the details, the only difference for the details is the orientation of all those hydrogens and carbons. So that's why these are stereoisomers. Okay, right. Let's look at another example. And by looking into the details, for stereoisomers, we can further classify them into cis or trans, as you see over there. Okay. Another way to classify them is classify them as enantiomers or diastereomers. So what are enantiomers? Enantiomers are the stereoisomers that are non-superimposable mirror images. Okay. Diastereomers are stereoisomers that are not enantiomers. So let's look at a more simple example. For example, if you have a tetrahedral shape, you have a carbon. All right, when you look at the solid lines, let's do FCl. The solid lines means they are in the plane of this surface. Now you see the wedge. Wedge means it's sticking out toward yourself. All right, let's use iodine over here. You see the dash. That means it's sticking away from you to the back of the paper. Okay, so if I look at a different structure, like you see over here, this is fluorine, now carbon, now you have chlorine here. So the one sticking out is also iodine. Okay. Then the one sticking to the back is hydrogen as well. It's not in the plane of paper. Okay. When you look at these two molecules, as I just drew over here, when you try to superimpose both of this, you cannot do that. Although the four groups attached to the carbon are the same, but you look at the details. These two are actually mirror images of each other, but you cannot superimpose them. Just like your two hands, they are mirror images of each other. But if you try to superimpose them by putting your thumb on the thumb and then the index finger on index finger, etc., you just simply cannot do it. So these isomers or stereoisomers, we call them enantiomers. For diastereomers, it's the stereoisomers that are not enantiomers, or the other stereoisomers, we call them stereoisomers. Okay. If we summarize everything, here's how you can determine the relationship between two organic molecules. First, you try to decide if two molecules are isomers. I do remember there is a trap. You do need to make sure those are not identical compounds, so you know they are isomers. Okay. If they're isomers, it could be constitutional isomers, which have the same chemical formula but different connectivity between atoms. If all the connectivities are the same, then they are the stereoisomers. Stereoisomers can be further classified as enantiomers and the diastereomers. Remember the cis trains, they have the same connectivity, so they are the stereoisomers. And cis trains, they are not mirror images of each other, so they are not going to be enantiomers. So the cis trains isomers, they are actually diastereomers. Okay, just to clarify that if you are a little bit confused. All right, next we're going to talk about so-called chirality. What is chirality? We actually went over this without specifying that. If you look at this example, look at the two structures it's drawn. You look at this carbon and the other carbon. Okay, both carbons are chiral, or they have chirality, because the four groups attached to that carbon are different. If any two or more attached to this carbon are the same, then they are not chiral. So chirality is generated by the different groups attached to the carbon. All right. So when you look at this carbon, again, this is the corner. So you have carbon. You do know you're connected to 1, 2, 3, 1, 2, 3, 3 single bonds. That's 1, 2, 3 single bonds. So you do know you have hydrogen surrounding that. So you look at this particular carbon, you have this group attached to it and this group attached to it and another big group attached to it, right? Okay, so that carbon has 4 different groups attached to it. So that carbon is chiral, all right? What about, then you might be asking, hey, what about this carbon? Is this carbon chiral? Is this chiral or not? It's not. The reason, we can look at that. For this carbon, if you write the details, it's connected to another carbon on the terminal, okay? We don't have to write the structures for the next carbon, but I'm writing it anyway. So this carbon is what we are studying, right? This carbon would need 2 more hydrogen attached to it, because you do not have 4 different groups attached to this particular carbon. So this carbon is not chiral, all right? But the neighboring one is actually. So chirality is generated by the different groups attached to the same carbon, all right? And most of the time, you would see the drugs being made in the pure compound of one enantiomers, but sometimes you do see examples of a mixture of different chiralities of the same compound. Okay. Let's look at one more example. Identify which of the molecules shown here are chiral. So if you look at this carbon, it's not chiral. Why? Because that carbon is connected to 3 same hydrogens, right? So this achiral or not chiral, this is achiral. When you look at this carbon, what about this carbon? This carbon is connected to a 2CH3 group, right? So they're the same, it's not chiral. What about this carbon? So what about that carbon? Oh, that carbon, is it chiral? Oh, so we look at the groups attached to that carbon. So this is a carbon over here. You have 3 single bonds, so you know you're connected to 1 hydrogen. Then you look at all the other atoms that is connected to it, and everything else, the 4 groups. So that's 1 group, that's another group, that's the big one serving as a group, and the hydrogen as the 4th group. Are any 2 of them the same? If not, it's chiral. If yes, then it's achiral or not chiral. So when you look at the 4 groups I circled, they are different. So that carbon is chiral. All right. So can you identify this one? Is it chiral or achiral? Oh, this is a carbon. Oh, you are not even attached to 4 different groups. It's 3, so it's not chiral. Okay. So what about this carbon? The same way, it's attached to 3 groups. Now what about this carbon? Oh, you have 2 hydrogens the same, so it's not chiral. And the next one, achiral as well. Yeah, and not chiral because you have 2 same groups. All right, what about this one? Oh, that is chiral. Why? Because when you look at this carbon, when you look at the left side, okay, it's attached in different order. When you look at the other way, and this methyl group. So for this particular carbon, as you can see, this carbon over there, that is chiral because you're attached to 4 different groups. Okay, I'm not going to discuss the other examples, but I hope you read through this carefully. Let's go on and summarize this. So for organic molecules, here's what we need you to know. First, you need to know different ways to represent an organic compound. You have structural formula, condensed formula, chemical formula, and line structure. Do know the details of the differences between each one of them. Second, organic compounds can have isomers. And isomers can be constitutional isomers or stereoisomers. Stereoisomers can be further divided into subgroups of enantiomers and diastereomers. So that's all about the organic molecules we need to know. We have barely scratched the surface of organic chemistry, but for the purpose of this general chemistry, that's all you need to know about these organic molecules. Now we're going to go on and talk about some biomolecules. We're going to start with the proteins. So proteins, we're pretty familiar with this. They are biological polymers made of amino acids. Okay, so amino acids, they are molecules that contain the amine portion. So what is amine? Recall that function group we just talked about. That's quite important. That means it's nitrogen connected with certain number of carbons or hydrogens. Okay, and you also have carboxylic group as the function group. If both function groups, you have the amine and the carboxylic acid function groups on one molecule, we call it amino acid. You also need to know the 8 essential amino acids. I do not expect you to memorize all of them, but you do need to know some amino acids are essential because they cannot be synthesized by our body. Do remember histidine is essential for infants, but not for adults. Let's look at the details about amino acids. Look at the structure. When you look at the structure, you do have the carboxylic acid. So what is the COOH? COOH is the same as the functional group as what we saw before, CO double bond connected to OH group. Okay, so that's so-called the carboxylic acid function group, if you recall. Okay, then on this side it's connected to a certain number of other structure, a carbon, for example. Okay, So that's carboxylic acid function group, carboxylic acid function group. All right. Then you have amino group over here. See? So you have amine, you have acid. So that's why this is called amino acid. These type of amino acid are called alpha amino acid. So why do we call this alpha amino acid? So alpha means the position of the amine where that NH2 is connected. So let me give you one more example so you understand this, what alpha position means. Alpha means the carbon that is neighboring the particular function group you're talking about. Here is the carboxylic acid. For example, if I give you a line structure for this carboxylic acid, okay, that's a carboxylic acid because you have COOH, which is equivalent of what I drew over here. That's carboxylic acid function group. So how do we name the carbons? The one that is neighboring the COOH group, that's called alpha position. Then you have beta, you have a gamma, you have a delta, etc. Okay, so if I put an NH2 group on alpha carbon, okay, that would be alpha amino acid. Okay, if I put the NH2 on beta position, that's also an amino acid, but it's not alpha amino, it's beta amino acid, okay, instead. So yeah, if I put the NH2 on beta carbon, okay, that's beta carbon. You see beta carbon here, then it would be a beta amino acid. Okay, all right. So that's some of the things you need to know. Let's look at the 20 common amino acids and group them into different categories by looking at the so-called side chain or R group. So what is the R group? R group, that is the group that is not the NH2 group. NH2 because it picks up one H plus. That's why it becomes NH3 plus. Okay, that's if you're wondering. And you look at the COOH, that H plus is picked up by NH2 in water. That's why you see this kind of structure, if you're wondering. All right. So you also take away this one of the hydrogen over here. Okay, you see everybody, you have the three same groups. If you have noticed, you see that amine group and a carboxylic acid group and the hydrogen. Okay, if you look at every structure, they look almost exact the same in those three. Okay, what the difference is so-called R group. That's the R group we're talking about. That's the R group we're talking about. Okay, so we classify the 20 amino acids by the R groups. So the first one, we call them nonpolar. Okay, the R group hydrogen is nonpolar. CH3, hey, hydrocarbons are nonpolar. We have learned this in general chemistry when we do the polarity of molecules. So hydrocarbon chain, you just see that. What do you mean hydrocarbon? I mean hydrogens and carbons only. Okay, hydrogens and carbons only. Okay, see almost all of this, you're going to see hydrogens and carbons as a side chain. Hydrogens and carbons, you see that? Okay, hydrogens and carbons, because this is benzene ring, it has carbons and hydrogens as well. Okay, all right. So the only exception is when you look at tryptophan, you do have an NH over here. That's one special case. Okay, you got to remember that tryptophan. We will not ask you to memorize the structure themselves. We will give you the structure, and you must be able to recognize and classify them as nonpolar, polar, acidic, or basic. All right, we're going to go over those as well. Okay. Another one, kind of special, for methionine, you have a sulfur group over here, but the others, they're also hydrocarbons. So if you have hydrocarbons only as the side chain, of course it's nonpolar. But for tryptophan and methionine, you have some heteroatoms. So those two probably you got to remember. You have sulfur here, and you have an NH over here. That's kind of special because the major portion of this tryptophan side chain, it's nonpolar. So the whole thing is very nonpolar. The second type, of course, is going to be polar groups. For polar groups, Usually they contain OH and some other hydrocarbons. OH, you notice that, and some hydrocarbons. And SH, which is so-called a thiol, okay? It's also a polar, and the hydro groups is OH, right? Then non-polar portion and the polar portion. And the carbonyl, as you can see, carbonyl group, CO double bond is very polar, okay? So this one, asparagine, is also polar. So you see CO double bond, okay, CO double bond or OH, or you have a SH, okay? Again, we will not ask you to write the structures of each amino acid, but if we give you the structure, you should be able to classify them. The third type, according to the R group or side chain, is the acidic side chain, okay? So we call this acid group. Again, this hydrogen is lost, okay? So this one, you had a H plus over here, it would be a carboxylic acid, right? But because in water, actually, this hydrogen is deprotonated, or H plus is taken away. So that's why you see, you do not see the hydrogen over here. Similarly, see, that's very easy. For the acid chain, it's very easy. For the base chain, base side chain is also easy. Basically, you have some different amine groups over here. You see that amine means nitrogen connected with certain number of hydrogens. Okay, you see amine, you see amine. Okay, this is arginine. You have a 3 NH groups over here. Okay, do remember for these 3 ones, don't confuse that with the tryptophan. You look at tryptophan, you see tryptophan also has NH over here, but this is not a basic side group. Okay, and you have NH2 over here. That's not basic. Okay, so because you are connected to actually with CO double bond or carbonyl, so this does not make it basic. Usually, if we say, oh, if you have amine type, oh, this is actually amide because it's connected to CO double bond or carbonyl connected to NH2, and that's actually a so-called amide. Okay, so this is not amine. Okay, that's why it's not classified. You look at these groups over here. Look at these groups over here. That's why these are not classified as basic. Rather, they're classified as polar side chain. Okay, all right. You might have also noticed one thing, as we mentioned, is on the structure of the amino acid. Sometimes it's written differently. Over here, the first one, you do see the NH3 has picked up 1 hydrogen. And for the carboxylic acid functional group here, the OH is retained, right? But in the physiological environment or in our body, the amino acid is going to exist in such a way so this functional group for the carboxylic acid will have the hydrogen being lost to the NH3, which results in a negative charged portion and a positive charged portion sitting in the same molecule. If you see this case, then we call this ion, so-called Zwitterion. That is a molecule that has both the positive charged and the negative charged groups in this structure. So that's this Zwitterion you see over here. The first one is not, this one is not Zwitterion. The last one is not Zwitterion either. The first one is only positive charge, that means it's cation. The third one is anion. The one in the middle is so-called Zwitterion. As long as you know the concept, it's fine. Then the next concept, you do need to understand when the pH in which the amino acids exist is different, then the different species, as you see over here, the first one, the second one, and the third one, they will exist in different amounts. Okay, we're not going to go into the details. You just need to understand pH will affect the relative amount of different species of the same amino acid. Okay, they will affect the different amount, the ratio of different compounds. Okay, that will be existing in the same solution if the pH is different. Next, about the peptide. Peptide are actually made from the amino acids. For example, if you have a valine and a serine, okay, the carboxylic acid when it reacts with the amine portion of a different species or the same amino acid. Okay, if you combine, you see the blue circle here. If you lose water, then the carbon and the nitrogen, they are connected, and the newly formed bond, we call that a peptide bond. Okay, so when you look at in this structure, this CO double bond and the NH, they are indicated as the peptide bond. But if you just indicate the CO as the peptide bond, it's also correct, because CO bond is what is actually connected. So if you write the whole thing as you see over here, this thing, they highlighted in blue, if you write that as the peptide bond, that's correct. Or if you simply say the CO bond, not CO bond, I mean the CN bond, CN single bond, as you see over here, if you indicate that as the peptide bond, it's also correct. Right. Okay. Let's look at some of the functions the proteins can actually perform. So some of them are just for the structural formation. Some are serving as catalysts or enzymes. Okay. Some are hormones. Some used for signaling. Some for the immune purpose. Some for storage. Some for transport. These are the functions, but no details are needed. Okay. Proteins have different level of structures. Actually 4 different level of structures. The first level is so-called primary structure. Primary structure simply refer to the sequence of the amino acids in the peptide chains. Remember, when the peptide bond is formed, the CO functional group and the NH is connected after the loss of one water molecule. Okay, the individual amino acids in the chain after the loss of water, they are so-called the residues. Okay, the proteins contain the same number of residues, but different identities or sequences. They will have different properties, and the specific sequence is referred to the primary structure. Okay, the secondary structure describes the geometric patterns by the segments of the amino acid chains. So one common pattern is the alpha helix, as you see on the left side. Okay. Another feature is the beta sheet or beta pleated sheet. Okay. What about the tertiary structure? The tertiary structure involves the interaction of different portions of the protein via the ion-ion or dipole interaction, or van der Waals interaction, or sometimes covalent bonding. So this will result in extended 3-dimensional features that are responsible for the protein's biological activity. When you look at the examples of tertiary structures, include this disulfide bond forming between the cysteine groups located in different regions of this protein. So in this structure, some of these places, they are some portions of the structure, they are pretty close, because for the side chain, for example, of the cysteine group, which has a SH, okay, they can actually undergo a reaction that will connect the 2 sulfurs. Okay, that's why our hair becomes curly. Okay, so tertiary structure involves the intermolecular forces, which results in different 3-dimensional features. What about quaternary structure? Quaternary structure refers to the larger overall structure that functions as a single unit. So for some proteins, they might have several units, but how do they work together? So this representation would show you how the 4 different units work together as a single unit, and this whole overall structure is so-called quaternary structure, which gives you a full picture of how something works, right? How does a protein serve as a catalyst or enzymes? Okay, we have different models for that. In this model, you see the active side as indicated over here. Okay, all right. Then you have the substrate. When they react together, you form an enzyme and substrate complex. Okay, unlike in the inorganic catalyst, as we discussed in kinetics, enzymes, they're highly selective. They must match structurally for this reaction to proceed. And this selectivity is related to the three-dimensional structure of the enzyme. I do remember enzymes are made of proteins. Okay, the structures for the specific protein that function as enzyme is really important for the selectivity. For enzymes that are highly efficient and selective, do remember this kind of selectivity can extend not only to the particular kind of substance, but also to a particular enantiomers. Okay, even for some, as we know, enantiomers are mirror images of each other, but not the superimposable. Okay, they look very similar, but enzyme might react with one enantiomer, but not the other. Okay, what about inhibitors? Inhibitors, they are the compounds that will diminish or destroy the ability of an enzyme to catalyze a reaction. That's so-called inhibitors. As long as you know this concept, it's okay. All right. There are two possible ways that an inhibitor may change the catalytic activity of the enzyme. So when you look at the first case over here, the inhibitor actually binds to a certain portion of the active site. So after the binding to that particular site, this prevents the substrate from binding and thereby preventing the reaction. So this is so-called inhibitor. Now let's go on and talk about the next type of biomolecules, carbohydrates. Okay, carbohydrates are a class of compounds having the generic formula. You have a certain number of carbon and a certain number of H2O. That's why they are called carbohydrates. Carbo means carbon, hydrates means water. But do remember, they are not a simple combination of carbon and water. It's just a formula. Looks like it's carbon and water. Okay, all right. Here's a structure of alpha glucose. Okay, in this alpha glucose, you have the open form and the cyclic form. Okay, so I'm going to explain a little bit to you why the left side is called alpha glucose. The right side is called beta glucose. Okay, alpha or beta is comparing this carbon 1 OH with the carbon. Did you see carbon 5 over here? On carbon 5 you have CH2. This is CH2 and OH, right? So you are comparing the position of the carbon 1 hydroxide or OH group, okay, with the CH2 OH, as you see over here, CH2 OH. That's a CH2 OH. If they are on opposite side of the ring, then we call this alpha. You see that? This is down. This is CH2 group on the top. That's up. That's why this is called alpha. Look at the right side. Okay. Look at CH2 group over here. CH2 group is traditionally always on the up position, as you can see over there. All right. Then you look at that. This OH is also up. It's on the same side of the ring. Okay. And remember, it's three-dimensional. You got to have some imagination to know this, because this hydrogen, as you can see, it's down. So this OH is actually up. Okay. It's on the same side. When you look at relationship between this OH and CH2OH, they're on the same side. If it's on the same side, it's called a beta glucose. Okay, if you're wondering why we call one alpha, one beta, that's how that works. This next example is fructose. Okay, as you can see, compared to this CH2OH group and OH group, if the OH is the carbon that is connected to that oxygen. Okay, this is the carbon. This is carbon that is connected to this oxygen. Okay, you compare that OH as you see over here. So this two relationship, one is up, one is down, different side, that's alpha. Okay, CH2OH, and then when you look at this OH group, it's on the same side, so we call that beta. So that's how we define alpha and beta. In this slide, you see a disaccharide being formed. Disaccharides or polysaccharides are formed with monosaccharides like glucose, fructose, when they combine via so-called condensation reaction, in which you undergo a loss of a small molecule like water, as you can see over here. Okay, that's so-called condensation reaction, in which you lose a small molecule. It could be H2O, could be HCl, etc. The resulting COC bond, this is carbon, this is oxygen, this is a carbon. So this is COC bond, as you see over here, this bond. Okay, that's so-called a glycosidic bond. You can also form a polymer if many monomers condense together. For example, you might form so-called alpha 1,4 glycosidic bonds. Okay, I don't want you to know the details, but it might help you if you understand why this is called alpha. Okay, because again, look at that. Look at this CH2OH and look at this position. It was originally OH, right? They are on different side of the ring, so that's why this is called alpha. Okay, for beta, look at the OH, CH2OH, and this O, which is supposed to be OH. Okay, the H is not written, right? The H is down, so the OH, original OH, is up. It's formed by condensing the beta glucose monomer. That's why this is called a beta 1,4 glycosidic bonds. Okay, no more details about why 1,4. They are related to the position where these OH groups are connected. Let's look at the next type of biomolecules. That's the lipids. Okay, when you look at lipids, they are simply the class of water-insoluble compounds. Okay, for example, glyceride, they are the lipid containing the ester formed. Remember, what is the ester functional group? We're going to review that in the lipid. It's the ester formed between fatty acid and the alcohol, glycerol. Okay, so what is the fatty acid? The fatty acid is just it's the salt of the glycerides containing primarily saturated fatty acids. Fatty means it's fat, it does not dissolve in water. So the fatty portion is actually the hydrocarbon portion. Okay. For the oil portion, it's similar in structure, but with unsaturated. What do you mean unsaturated? Which means you have a double bond, a triple bond. Okay, let's look at one specific example to clarify this. So that's so-called glycerol. You have 3 OH groups, which can be used to do a condensation reaction with the so-called fatty acid. Remember, this is the acid. Why do you call fatty acid? Because this portion is hydrocarbons, so they are insoluble in water. 3 molecules of fatty acid condense with the that's the glycerol portion. Remember, waters are lost. Okay, so waters are lost. So you actually have 3 molecules of this, then you lose 3 waters here. Okay, the compound that is formed, that's called a glyceride. That's a type of lipid. But yeah, we have other different types of lipids. Okay. All right. Saturated fatty acids and versus unsaturated. I want you to read this on your own. You don't have to memorize it. The key difference between the saturated and non-saturated, as you can see, is whether or not you have double bond or not. Here, no double bond. Here, you see double bond. Here, you see double bond. Okay, all right. Phospholipids are used for the structural formation for the double layer. Okay, all right. So that's another type of lipids. As long as you know, phospholipids is also a type of lipid. That's fine. Cholesterol, when you look at the structure, you have one OH group and all the other portion, it's hydrocarbons. So apparently it does not dissolve in water. It is also another type of lipid. Right. Now, the last but not the least, we're going to talk a little bit about RNA and DNA. RNA and DNA, they are made of the monomers that is called a nucleotide. A nucleotide is the monomer unit in which both the RNA and DNA are comprised. In the structures, as you can see, a nucleotide is made of 3 different portions: phosphate, sugar, and the base. As you can see, the 5 nitrogen-containing bases found in nucleotides are illustrated over here. All of them contains multiple nitrogen atoms in the aromatic ring structures. All of them, except for adenine, also contain one or more carbonyl groups or CO double bond. Right? The names and structures are shown in this slide. I don't want you to learn the details about this, but the main feature, as you can see, they have the ability to form hydrogen bonding between different pairs. We will discuss a little bit later. As you can see on the left side in this DNA structure, the nucleotides that represent the structural unit for the DNA and the RNA, they were identified previously. Remember, one nucleotide is composed of 3 different portions: phosphate, sugar, and the base. Okay, the polymer formed from the combination of the nucleotides, they are DNA or RNA if you have different sugar. Okay. For the pairing of the bases, it's based on the matching of the hydrogen bonding forming ability. As you can see, adenine is matched up with thymine because of the hydrogen bonding that can be formed in between, as you can see over here. All right, so A is matched up with T, and G, guanine, and the cytosine, they are matched up because of the hydrogen. You can form actually 3 hydrogen bonds between these 2 molecules, and these hydrogen bonds are responsible for the DNA structure, double helix structure. That's about it. So we still have 3 more slides I want you to read on your own. Let's summarize everything. As you can see, we have covered a lot of topics. It's virtually impossible to know all the details about everything. But what do we need to know? I want you to spend a lot of time studying how you can represent the same organic molecules differently. For example, you have the structural formula, you have the condensed formula, you also have the line structure, you also have the chemical formulas. That's one of the main things that will be tested. Secondly, for the different biomolecules, pay special attention to the proteins. Okay, proteins are formed by condensing amino acids. You have 20 different amino acids. You should be able to classify them into 4 different groups according to the side chain. For the different side chains, you can classify them into nonpolar side chain or nonpolar R group, polar R group, acid or acidic R group, base or basic R group. For the other types of biomolecules, just a briefly do a survey type of a study that's going to be good enough. Probably we're not going to test too much for those other biomolecules. All right, that's it for today.